picking up the viral chapter. There's a couple of really important things. The major thing that's going to be on the exam, more than anything else, is about the prions. But so I do want you to, but you have to be familiar with the other stuff that's here. So allow me to show you where we were. When I get back to that PowerPoint, and that's this. In general, infection, and we'd go over this again. In general, infections, sort of like traffic. You have an incubation period. You have the illness itself. Sick there. I have recovery. In general, if it's the kind of one that comes on fast, it goes away fast. If it comes on slow, it goes away slow. That's a generalization, but it's a fairly accurate generalization. The longer it takes to hit you, the longer it stays. There always are going to be exceptions to that, but in general, that's very true about biological things. You know, I mean, the difference between the nervous system and the endocrine system. Endocrine changes are slower than nervous system changes. Nervous system changes go away much more rapidly. So now the interesting thing, for the most part, when we get to how they spread, depending on it, usually about halfway through the incubation period, this is the area where you and I would call contagious or infectious. You don't have to have it to be infectious, and that's like we talked about norovirus. I mean, you're barely symptomatic, and in reality, you're starting to shed the virus. And it's just not for virus infections, but apply to bacterial infections. From a standpoint of understanding health implications and public health implications, for many of you going into healthcare, this is really important. So you begin to know when somebody potentially, when you evaluate them or you see them, what stage they're in. So I think that's kind of important. And we reiterate this, the chapter, once we get done with this, and effectively when you come back from break, that's where we're going to start. Really the second half of the course is very, very clinically oriented. We start to look at how they spread. We look at antibiotics and various kinds of anti-infective things, antivirals. And then basically it's all diseases after that. You must be doing the antibiotic lab this week, correct? Incorrect? I saw a big, you don't know what you're doing. You know what you're doing in lab ahead of time? Huh. All right. Well, I saw they're making up plates for that, so it looked like you're doing that. Because they were in the lab. I was just in there earlier this morning. Nevertheless, when we talk about an acute infection, again, most of the time it's a relatively rapid onset, a relatively short duration, and it's associated, and the virus particles are somehow being released, whether the cell is bursting or whether they're being shed, sort of popping out. I mean, and so there's a lot of different terminology we use for how virus particles escape. So budding or shedding, and if you remember that previous illustration, like this, picking up some degree of membrane on their way out. They may have gotten it here, but most likely they got it from the cytoplasmic membrane of the bacterial cell. And see you later. Okay. The difference is we have persistent infections. When you talk about persistent infections, that includes HPV and herpes, because effectively they're there for a lifetime. Does it mean they're ever going to affect you again? No, that's different. And you may or may not have symptoms. And again, this applies to bacterial infections. A lot of times we see people who are what we call asymptomatic carriers. That is a big problem. A very, very large problem when we get to see this. Someone who carries the problem but doesn't demonstrate it. And so, and again, HIV again, that's the part about this. It's kind of both. You have an acute phase, and then the persistent phase can be really problematic. So there's different ways, and this is a lot of this, and it's a little confusing terminology. Persistent infections, when they talk about either chronic, and that's continuous production of low levels. And again, these are these asymptomatic carriers. So we actually have people with both hepatitis B or hepatitis C who conceivably can be shedding particles all the time. HIV is the same way. I think I mentioned it to you, but when someone's finally diagnosed with human immunodeficiency virus, one of the first things they look at is what they call the set point. The set point is how many viruses are in there percentage wise, and they can assess this at the time they're diagnosed with the disease. Assuming then they are being treated appropriately, that's a very significant factor as to whether or not, just how likely contact is, of an intimate nature, is to potentially infect the other person. Okay. And there's just so many nuances to this, and depending on what you do for a living, or what your lifestyle is, or who you interact with over the course of your lives, and there's a lot of variation, everybody. These are really useful things to know, much less being in an occupational setting, being the kind of work that you do. So latent infections, it's silent. That's the classic cold sore. That's the difference between chickenpox and shingles. It's completely latent, okay? And then it will reactivate. It doesn't, again, the difference between herpes, cold sore coming back, for argument's sake, a wart coming back, for argument's sake, or having a totally different presentation, that reappearance of the chickenpox virus, not as chickenpox, but as shingles, a different kind of, totally different visible presentation. They all count the same. Those are still reoccurrences of latent infections. It's the reoccurrence of what's causing the infection that's a significant factor. And I mean, these are some examples. I told you a little bit about cytomegalo. Okay, typically it's not something you ordinarily would see unless you're dealing with immunosuppressed patients. So I don't spend a lot of time on it. But this is a classic example. I shared with you that my first wife, I mean, she had it when she was your age in college, and did I not share that with you? Yeah. And then she had it when she was an adult. An adult was much worse. And again, this one happens to be associated with certain kinds of Burkitt's lymphoma. Is an oddball. You generally see it in Africa. It does affect glandular tissue, but I mean, there's different kinds of nose and throat cancers that are associated, rarely, but they do occur with Epstein-Barr. Hep B and Hep C, big time players. Again, I mean, if someone has a persistent carrier of hepatitis C, I am really careful. Unlike someone who's HIV positive, that random needle stick with someone who's Hep C positive, you can get hepatitis C. And so that what you're armed with, look, many of you are signing in to treat people. It's not something we selectively refuse. Oh, I don't want to treat them. And people did that years ago, and they didn't understand it. What I'm trying to share with you, and many others will share the same thing with you, you're armed with the information of what you have to do. Okay, I got to be super careful. Do I have to double glove? Maybe I do. Should I wear all that gear that they have outside the door, the gown, the glove? Don't play fast and loose with those rules, because it's not just about protecting yourself. It's about protecting other patients, your colleagues, your staff, etc., etc. This basically, everybody, is the Sermon on the Mount for today. You know, this is what you chose to do. There's a certain element of risk that's associated with it. The only way we can cope with elements of risk is to understand how to minimize the likelihood that we will be infected by it. You can't avoid it. You're going to get I keep telling you, ask anybody who's been around and been treating people for a long period of time. Anything happens. Weird stuff happens all the time. Moving on. HIV, obviously, it's mixed in the sense it's effectively latent, but if it's not treated, we call it a slow virus, because over 2 to 10 years, it will rear its ugly head. And at that point, your immune system is going to collapse. And at that point, you're going to die sooner than you should be dying, etc., etc. I mean, all of these are different types of things. I mean, this is what I'm terrible. Artist, typical flu, depending on how severe it is, it could be a lot longer duration, but yeah, typically the time is in days. Roughly, people are infectious when they approach having full blown, and they're less infectious afterwards, but it's still there. The fact that I'm feeling better doesn't mean go out and do stuff. Hepatitis B or C, If it's not treated successfully, and there's a lot of people who will be a carrier, it doesn't mean they are. But this would be when you hear someone has a chronic hepatitis B or C, you have to be very careful, particularly most of you are immunized for B. C is the one I think you worry about a lot. And then here's this typical latent infection, okay? Typical herpetic infection, cold sore number 101, non-infectious until, and again, once you get to about here, it starts to be infectious again. The difference with AIDS is it's almost like you can draw a line this way. You have HIV. HIV is you feel like you got a bad case of the flu. There's no. It's when we get to genitourinary diseases, it's not like there's a specific sore or a specific rash or some obvious thing that you can identify. That's the problem. Some people hardly get sick at all. And it takes a while for them to get sick, so they may have had extra contacts, depending on how, again, lifestyle. I don't want to get preachy about it. It's your life. Do what you want. I'll talk more about it when we get to genitourinary diseases. Just try to be as careful as possible, realistically speaking. And I'll say this again: if you choose a more perhaps a less cautious lifestyle, then do yourself a favor and keep getting checked on a regular basis. Okay. Now, moving on. Yeah, I know this is Grandpa telling you, but I had this discussion with my kids. So cold sores, I hated saying that. Cold sores, here we are, and they usually have one for. Let me get the age. It kind of goes like this: it's going away, and gradually over that 2 to 10 year period, then all of a sudden it aids, and now it kind of looks like that. Gives you the categories. They're just showing you typical location, ganglia of sensory nerves. This is the trigeminal nerve. Okay, and there's a lot of things. The virus integrates with the host cell's chromosome, can't be eliminated. Later can be reactivated. There's a lot of things that reactivate it. When we talk about chickenpox and shingles and herpes infections specifically, we'll talk more in the concept of disease. This is what's on the board. This is important because to understand it isn't just viruses. There are two factors that you have genetically that are there. And I read this presentation. I was doing this presentation, and there's a wonderful book which is not an easy read. I'll write it down here. I won a Pulitzer Prize in probably about 2010. And basically, this author, Emperor Bull Mountains, certainly find it. You can get ebook. I read most of my books. I like more my iPad. Well, the Emperor of All Maladies. He basically, this author, I won't even say it because it's so smart, but he was a doctor who basically took cancer as he sort of writes biography. So here we go. Proto-oncogenes are the inactive type. Okay? Presence of environmental factors, noxious fumes, gases, radiation. Take one, we've talked about a lot of them. Viral invasion, possibly. Lifestyle, smoking, drinking, all that kind of stuff. All of those inherently have they can effectively change these from their inactive form, proto-oncogenes, to oncogenes. Onco means tumor. So they become ones. Best way to think, and this is the way he discussed it in his book, think of these as being the accelerator. It's almost, when this happens, they go from inactive to active. It's like your accelerator is stuck. That ain't good. It's going to keep going faster and faster. It's kind of doing it to the cell side. The other thing that protects you, so normally these are inactive. We don't want them active. What we want to have active, and they are, the tumor suppressor genes. Those breast cancer genes, so this is not the example where really they get impacted. Those breast cancer genes, BRCA1 and 2, which a lot of people know. Okay, and you certainly should get an A and B. Those are the category of tumor suppression. When there's a fault in them, that's why folks are pushing so on the breast cancer. So they are normally active. Again, under the same circumstances, they can become inactive, and therefore your mechanism to stop cancers is compromised severely. Think of those as the brakes. So what happens if both of these are effective, you got an accelerator stuck and you lost your brakes. You're going to crash into something. Effectively, that's what's going to happen. I'm trying to use a relatively easy to understand analogy with this and let you know that this is a significant issue. That's what happens. This is more contextual. I don't think there's a question like this on the test, and I don't want you. I need to explain stuff to you, okay? And when I say it's not on the test, it doesn't mean, well, I forget about it. It's important. I led the wrong lifestyle for, I don't know, 40 years, 2 packs a day. Thankfully, I didn't drink. If I drank, I wouldn't be giving this lecture. I'd be dead. Because I was close, really close twice. Once before I was 50 years old. Understand that. And the award for the smoking, you all heard this was bladder cancer. Okay. I'm not telling you, live your own life the way you want to live your life. But understand the risks that are associated with it. Okay? Just saying. Okay. I mean, individuals do those things. I mean, I'm thoroughly surprised that all 3 of my kids have reached 40. So, to summarize, cancers, malignant growth, metastasize means they can spread. This is a separate discussion. Benign ones can damage you. Benign doesn't mean can't kill you. Brain tumors, 90% of them are benign. But they're occupying space, squeezing your brain. You get the idea. We talked about this in A&B. So proto-oncogenes stimulate cell growth and division. Tumor suppressor genes inhibit growth and division. Mutations, uncontrolled growth, either way. So, and again, multiple changes means multiple factors. The oncogene is the one that's been changed to promote uncontrolled growth, the definition of cancer. Okay? It could be a virus, chronic infections, part of why HPV, why that's really the poster child. You should be familiar with these, not this one, not the HTVL1 from effectively it's a relatively rare thing. But the ones that really impact you more than anything else, Epstein-Barr, well known, hepatitis B and hepatitis C, HPV certainly, that herpes type A, Kaposi sarcoma. Understand those are ones that were just well known to be associated with viral infections. This is a virus that's directly leading to cancer. That's important. And they go through all of this stuff. And I don't find the slides to be that helpful. I don't find this to be so helpful. Why we don't do virology here in the lab, because of viruses. There's layers of protection. That's a fairly generic bacteriology lab. Some stuff we have to be more careful with than others. But by and large, we're not dealing with something that, if it lets loose, really would have a more significant problem. So that's why we have a BSL, biological safety level type 1 lab. Okay, it's modified because we do use some agents like Staphylococcus aureus that conceivably could cause infections. Pseudomonas aeruginosa, Proteus vulgaris, etc. Okay, and we'll look at the changes that are there. These are some of the things. Remember I talked to, see, viruses don't behave the way bacteria make toxins. Viruses damage cells. Here's one. That's when we talked about RSV, fusing cells together. Big trouble. Inclusion bodies. That's the genetic material incorporating with the nucleus. That's how we diagnose some of them. We see inclusion bodies. And that usually tells us, oh, there's potential for the DNA to be altered. Just gives you some idea. Not on the test, but important to know. The syncytium is a word I'd be familiar with. going forward. We're not going to count them. Okay, we're not going to test them. That's lab stuff. I've never taught this. I mean, it depends on the field you're going into that you may become familiar with this. I don't know enough about plant viruses other than they're very significant. Okay, very, very significant. Particularly, it's kind of things we look at viral infections. They really affect crops, costs, prices, availability, etc. Okay, and I don't know enough about viroids. Again, plants. Okay, single strands of RNA, but prions, which is the last topic for this chapter, and the last topic for the exam. Thanks. You don't have to thank me. I might put you guys to sleep. I know. They went to NBA school. You said they should agree with college. That tells you. Prions, protein infectious particles that you can see there. They're proteins that have been misfolded. They're just protein. Slow, fatal human and animal diseases. A lot of people think it may all be the same misfolded protein with subtle variances. Okay? And there's a lot of interesting nuances to it. Usually, not always transmitted within species. And cow disease would be a perfect example. And that was the first one we heard about back in about 1986. That's an example of transmission. So it affected cows. Okay. And if you're, I mean, one of the, England is a country that's a very large beef eating country. They are beef eaters 101. Okay. The cow, some type of product, most likely it started in cows. And the ingestion is the problem. We think it's ingestion. Okay. And again, I would never, I think that my frame is we think. We don't know, so I really can't tell you. But what happened was people started to die and they identified it because they saw things that were associated. This was fatal. It took, from the time people started to recognize it, slow, about 2 to 10 years, and they saw a specific kind of neurodegenerative change called spongy encephalopathy. Encephalopathy means brain disease. Spongy, or really the appropriate term is spongiform. When they did it, when they looked post-mortem, before we had a lot of technology and really had MRIs that could really identify these things, the brain looked like it had little, like you get a big sponge at the beach or one of those, it's got holes in areas where there's no material in it, little gaps. That was a classic form of it. Interestingly, they saw other things, irregularities in proteins that were tangled and irregular, and aspect particularly in nerves where the fibrils, the tiny little passageways that took materials back and forth along the nerve that we had talked about a little bit in AP1, they saw that they were tangled. These are hallmarks, interestingly, of Alzheimer's disease. We're pretty sure that about 5% of Alzheimer's is due specifically to prion invasion. Many, many years ago, I was teaching CCAC. One of my students, her husband was a researcher at Pitt, PhD researcher at Pitt. His research was specifically looking at prions. To give you an idea, so this is not something that isn't being studied aggressively. But for our purposes, these are the things you need to know. I think most likely it is the ingestion of neurological structures, like eating brains. No other way to describe it. Okay. I have not had any brain injuries. I try to avoid it. No, but the reality is why we think it happened to cows. Years ago, before it was made. The fact that it is illegal does not mean it's still not done. For interesting, okay, what happened with these cows? When a cow would when they would take a cow and slaughter it for meat, they'd probably take the hide and use it for leather. What's left of the cow? If they don't just dispose it, basically they grind it up, bones, brains, interior, nerves, grind it all up. It's called bone meal. And they put that back into the feed for the cows. Now that is illegal to do. There's a lot of distrust. I mean, if you're speeding on my way to work, I went over the speed limit. I'm sure I went 66 and 65. It doesn't mean that people don't do it. For instance, in Japan, who are very fond of meat, make wonderful and have wonderful meat called Kobe style meat or Wagyu beef. They will not take beef from our country or from Canada. We'll eat theirs, but they don't want to, they don't trust the process. So they used to do. They used to put that into all the food. For years ago, they changed bone meal. They thought they were reinforcing it with a lot of proteins, not realizing the potential problems with it. I mean, if you look at dog food today, that's not something that they put in. But what do we know about it? Multiple species of animals, always fatal, 2 to 10 years, spongiform encephalopathy. It is not associated with cancers. Not that we are aware. So these are just some of the names. Okay, kuru. This is why we thought it was consumption of brain materials. They found evidence of these prions in a disease called kuru in cannibalistic tribes whose bodies had been preserved based on the way they were buried or found. They could take reliable samples of brain, and again, they all had the same thing: spongiform encephalopathies, cattle, deer, okay, various kinds, cats. We get one that is inherited, which is unusual, called, it's pronounced Creutzfeldt-Jakob. When you hear about it, they'll say CJ disease. I'm not going to ask you the names of the disease. The one you should certainly be familiar with is mad cow disease or bovine spongiform encephalopathy. But there's a lot of these, and they may be, some of these we know that there's inheritance, so maybe there's a genetic trait that's involved in the way the proteins are. That's what most people would expect. And they demonstrate to you the difference. Nerves die, holes. You can see the holes here between normal and brain tissue. We need to have postmortem samples. Now we can identify it on things like MRI scans, typically MRI with contrast. Gives rise to a general term for all prion-based diseases. This is just trying to demonstrate how the misfolds occur. Not a big deal. Thus ends this. I know you're ecstatic. Okay. What I am going to do, and I'm working on it. So far, I have 15 questions. I'll probably get 5 to 10 more, and send that to you, which will be due after break as a take-home vehicle to try to assist in as a grade booster. So don't ask me why. I'm guilty again, because for some reason I like you people, the vast majority. Those who chat constantly sometimes attract my ire. Yeah, I know. I'm shocked. Yet's coming. So I will show you. I will endeavor to demonstrate to you an area where you can find some resources from what I'll be presenting. I'm going to talk about it a little bit today, just to introduce it. So this is your thingy, genetics and metabolism. I'm going to talk about the metabolism's hard. I'm trying to do a very generic presentation. So these are different PowerPoints that are associated with this. Okay. Just to give you an idea. So, the big factor, let me see if I can get one that's useful. They're both chapter 8s. How convenient. Okay. it's just remarkable. The distinction, to understand genetics are a difficult topic to explain. I try to keep it basic, okay, comparatively. This is the classic example. This lady eventually was awarded a Nobel Prize, which is great. Finally, a woman in science and medicine getting a Nobel Prize. It's all about corn, and that corn that we typically see in the fall, now called Native American corn. Not politically correct. So normally I get a kick on that. Who do you call? Go ahead. I know what you call. You just nevertheless, she noticed patterns in this very uniquely colored corn peony. Okay. And she concluded, because they were identical patterns, but they moved around and not a pattern that could be easily identifiable, but they always looked the same, just in different places. Okay, she figured out it was genetic. She published her research. Of course, typically nobody believed her. Okay, finally, the additional research, and she was awarded only 33 years later, and she termed them transposons. Transposons known as jumping genes. And there's a really useful animation that I will show you that plays well with this. But I'm not going to do the animations today. I'm going to do the animations on Wednesday, just to let you know about that. So that's the first thing. This has to do with different ways that genes move around. This is one of 4 methods that not just for me, I'm going to give it to you take-home style, but these are questions that you could easily get asked on a licensure exam. These are the kind of things people have to know about why a normal staphylococcal infection can become MRSA overnight. To understand just how this happens and how it happens so darn fast. And why a lot of times we can't figure out why that antibiotic didn't work. Okay? That's an E. coli that exploded out all its genetic material. When it dies, it explodes out the genetic material. Remember, it could wrap around an E. coli 1,000 times. That's how tightly wound, how super coiled it is. Understand it fragments, okay? It fragments into little pieces. Those pieces can travel. They can be picked up and move along, okay? Just to give you an idea. And that's something we have understood for a long period of time, the ability for those little DNA fragments to move. The research was done, are you ready? Roughly 100 years ago, before we had modern technology, electron microscopes, freaking knew what a bacteria was all about. Okay? Just to give you some idea. And so the mechanisms that change are mutations, okay? They mutate all the time. They're genetically inherently unstable. And this is what we call horizontal or lateral gene transfer. Remember me telling you that that's like, again, magically, you have great athletic prowess. You can just touch her, and all of a sudden, we all be great athletes. Please. That's what it is in the same generation. Normally, our genes are transferred vertically, mom and dad to us to our offspring, assuming we have offspring biologically, okay? All those types of things. I'm not telling you anything you don't know already. So really that is important, but you have to understand there's a major difference. They're just showing some pictures about it. So these are terms you should be familiar with already. What your genetic makeup is if we studied your genes. It's called genotype. How it appears is called phenotype. So whether it's the classic ones, freckles, hitchhiker's thumb, earlobes, maybe you studied those. Okay, there are certain things we know, your blood type. All of these things, your blood type, how it is, the genetics, understanding specifically which genes are impacted versus whether I'm A pos or B neg or O neg or whatever. Okay, that's the phenotype. And yes, there's influences from other things. That's basic genetic parlance. Those are terms you do need to know wherever you're going, because people ask you about it. Phenotype is an important term. This confused students forever. Okay. Auxotrophs means they need growth factors versus prototrophs means they don't. So I try to stay away from that. I only do this important. You gotta really know what's important about. So when we hear, or ones that you will hear about, and these are things that you will have to become familiar with when you see a superscript like that, R meaning resistant. Okay, streptomycin resistance is that. You know, this is how you may see this on somebody giving you a test, not me. One, I don't do it, and two, I don't know how to make superscripts and subscripts. See, you got to break it. Okay. The negative, meaning they can't make that, so they need that. That's something you might see. I don't do that. I would write down, if I was asking you a question, you know, can't make tryptophan. But it's not a question I would ask you. And this is interesting. You and I, our parlance for genetics, and you all know this, when we talk about a trait, we use two words, dominant or recessive, right? We don't do that at all. We talk, the way we talk about animals, even though they have dominant and recessive traits, if we see a normal animal, we call it the wild type. So if we see a fox of an odd color, that would be a mutant. That's the term, and we use that for bacteria because we don't understand it. Then we look at it. The term that they would use would be the wild type, the typical one we see in nature, versus some odd that we would call a mutant. So the parlance is different, and the whole idea is that the genetics are different. That's why I spent that time talking about what makes the difference. We have pairs of chromosomes. Okay, one from mom, one from dad. Bacteria have that one giant circle chromosome. The beauty of having pairs of chromosomes is that we only use one when we're generating a protein. Remember, we only pick one. And so if the mutation is here, and this happens to be the chromosome that is expressed, does it matter? No. That's the beauty of dominant and recessive. Pairs of chromosomes, if you have two dominants, the dominant will be expressed. If you have two recessives, the recessive will be expressed. And dominant and recessive, or recessive and dominant, the dominant will be expressed. So a lot of times it doesn't matter. A mutation, everybody, is an error in DNA. So what is? So the question is that the error in DNA of any consequence whatsoever? A lot of mutations are what we call silent. Either the error in DNA is inconsequential, we'll still assemble the same protein, or in our genetic makeup, it may not even be relevant. Okay, if it happens to be here, the other dominant will cover for it. If it happens to be here, the other recessive will cover for it. If it's one of these, okay, then there's even a 50-50 chance it won't occur. It's pretty hard when you have our kinds of genes in pairs, ours, anything, the elephant, the mouse, you go, any animal. Bacteria, not so good, because if the mutation occurs here, it's one strand, it's most likely going to express itself. It doesn't have any cover. That's one reason. Reason number two, we have a great proofreading mechanism called DNA polymerase, and it's got Roman numeral 3. All living things have some kind of DNA polymerase. It's a proofreading. So typically, and you're going to see it in bacteria, they have something called mismatch. And so remember, it's all A's, T's, G's, C's, same thing. So typically, an A and a T, a G and a C. If all of a sudden you get here, there are two G's, something's wrong. What if you was muted? Good, so we got X. Working with the rest, say XP. Expert. That was good. So mismatch repair, which bacteria have, you'll go, oh, good. Uh-oh, one of you is out, you're going. It's a 50-50 chance. Okay, that gets it right. DNA polymerase 3 does the same thing. We're good. We're good. Oh, you're really the culprit. You're normal. You're genetically different. You're the A and you're the other A, but this is where it's supposed to be. It'll identify the mismatch and replace the appropriate one. It's much more specific. Having a 90% chance of getting it right versus having a 50-50 chance of getting it right, much better. And lastly, something I didn't do in A&P, we have what's called messenger RNA processing. In our cells and eukaryotic cells, messenger RNA is a big long strand. And in that big long strand, it's got little pieces. Maybe you heard the term before. We call them I's and O's. I's and O's. I's are called introns. I'm not going to say that. Okay? I beg your pardon. I's and E's. E's are called exons. We have a lot of DNA that's important, but not all of it codes for proteins. Okay? What happens in your cells, in the nucleus, before the messenger RNA is let out, it removes introns. They're short for insertion sequences. So this area goes away, this area goes away, this area goes away. The only part, two little e's, are the part, so it's much shorter. If the mutation happens to be here on one of the introns or insertion sequences, it isn't expressed. Those are three major mechanisms that our kind of genetic makeup has that preclude, that limit the likelihood of genetic. For that reason, bacteria mutate about 1,000 times more frequently when they replicate than our cells. We got all these wonderful safeguards. How do we mess it up more? When we do all those lifestyle things that are bad, or we're exposed to things that are potentially mutagens. Radiation, noxious fumes, some idiot who smokes like a chimney for 40 years, that kind of stuff. Got it? That's what I'm trying. That's why this is important. There's a lot. I mean, we really have to become ultra familiar with these things. My wife's looking for an activation code. Check. No, 30 minutes ago. I'll get in touch. That's why this stuff is important. And we're going to see as it goes on 3 different categories. It's really important. I'm going to go through them relatively quickly. The 3 categories of mutations are, again, we have mutations. We mutate bacteria. We make the cells more. Okay? Sure, there's a few mutations. If they aren't expressed, we can call them silent mutations. Don't matter. It's called a miss. These are terms you will have to be particularly going forward. I'm sure there's a question on this. Missense mutations mean one amino acid. Remember, tell me whether they're a chain of amino acids. Okay. That can have minimal influence or very significant influence. To suggest that it's not important what amino acid, the difference between normal hemoglobin and sickle cell hemoglobin is. Insulin would probably make insulin that was unitively functional. It has to be a kind of type 1 diabetes, genetic. And the other one, the ones that you don't want to get, called nonsense mutations. They mean you assemble it. I mean, I don't go into all the assembly of how we assemble proteins. Nonsense mutations means they encounter. We stop. We have punctuation. We assemble proteins. We have start codes and stop. They encounter us. Stop code prematurely. That means you typically are going to have proteins that don't get assembled properly. That's usually associated with big trouble. Okay? With other organisms, that very often is incompatible with life or fatal going forward, depending on when they occur. Okay, that's what I mean. So there's a lot. I mean, there's a lot of terminology in that. I used to incorporate with this 2-chapter unit we're doing now as a 3rd chapter. Retrospectively, it is too much. So I have changed for a number of reasons, and I'm content to know that you need to be familiar with it and to give you, an opportunity to augment your grade. I'm using big words. You're almost done with me for a while. Heck, you're almost done with me for good. Let me reflect on that. You always say, you're going to miss us. But you are unforgettable. And if RMU has its way, they're going to want you to be unstoppable. I know, I can't do it. Goodbye. See you Wednesday.